I guess we'll just wait. Excuse me, wait for everybody to get on here. We might not get very many people, but that's okay. What is this doing? Why is it rubbing on everything? What are you doing? Why are you going in there? You don't need to be in there. One more. Oops. Now we're getting some people. Building up a crowd. I feel like I am on like a YouTube live. I don't know if you guys ever watch YouTube lives, but like they'll sit there and be like, have some background thing playing, just waiting for people to get on. That's what I feel like right now. Little dog. All right, so I'm going to try to be good about watching the chat while you guys are typing into it. I was kind of not the best at last time. I wasn't terrible. I think I got to everything eventually, but yeah, I just forget about it. So I have it open on the side there so that hopefully I'll actually see it. For the most part, I'm just going to have this PowerPoint could open like this and not up on the screen big, because I can scroll through it and switch in between this. And if you guys have questions about like the study guide itself, I can switch back to the study guide a lot easier like this. So if I need to write something, if I need to write something on the actual slide, then I'll make it big. It's just easier to do it that way. But so yeah, so that's the plan. So let's get into it, I guess. So it's Unit 2 review. You guys know same layout basically as Unit 1. For those of you who are in any of my Tuesday, Thursday classes, please bring number 2 pencils. Tuesday morning people, you will definitely be taking it on Scantron. I don't know what's going on with that computer in there. I want to act that with the clickers. I will try to figure it out, but I just don't know right now, and I don't want to mess with it for the exam. And then anybody who's in the Tuesday, Thursday, like evening, early evening and late evening sections, you guys bring number 2 pencils just in case, because remember it was weird last time when some people were connecting and some people weren't. I know we eventually got everybody connected, but like I'm sure everybody's patience was wearing thin with that, and I don't really want to do with it, so be prepared for that just in case. So don't forget your number 2 pencils. Monday, Wednesday people, just yeah, you're using the clickers. No getting out of that. So that's how that's going to be going. I like that it uploads faster. Scantron's not so bad, but it is a little bit of a pain. And I don't know if you guys know this, but it's sort of reliant on us scanning it through the machine and it reading it properly. And if it messes something up, it's kind of hard to tell if it does. It's not like I go back and check every single answer that you put on there that it matches up with everybody's Scantron. That's the whole point of the Scantron, so I shouldn't have to. But anyways, it doesn't work ideally a lot of the time. So that's why I tried to do more of the digital stuff, but technology has its own problems, right? Unfortunately. So remember that if it is in the lecture slides, I have to, warn you guys, it is fair game. If you guys have worked on the study guide, it should have gone through everything and understood the questions and all of that and done your definitions as well. You should be covered for this exam. I write my study guides. They cover all the material that I feel like you should have to know. The test is going to only test you on parts of that. But the idea is that the test, should be covered in that. So, well, I say it should be, it will be, I wrote it based on the test. And the other stuff that I feel like you should have gotten out of the lecture as well. So hopefully you've learned everything and you'll be prepared for anything. That's the hope there. So if you go through the study guide and you have something in particular that is bothering you, please let me know. All right, so I'm just going to go through a little bit of big topics of what we covered in each chapter again. And if you guys have any questions about any of it, ask in the chat or ask, through the audio or whatever, and let me know so that we can go over it in more detail. So chapter 7 had to do with microbial nutrition, essentially where microbes are getting their energy and their nutrients from. And we have all these different terms that we had to apply to them. Yeah, the main nutrients, the main elements that we were concerned with. Prepared me for that. I'm so sorry. Guys, what? Jesus. Okay. I'm sorry. I should scare me. The main elements that we're concerned about are going to be the CHOMPS elements. Okay, so that is carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. So those are the ones that we're going to be concerned about the most. That occur the most in the cell. That's why we're concerned about the most. All cells need to have that in them in order to function. So we have the photo whatevers and the chemo whatevers. We had the autotrophs, we had the heterotrophs, and all that. So if we were talking about photo or chemo, photo autotroph, photoautotrophs are the ones that their energy comes from photo, which is light, and auto is they are the ones that are going to be making their own organic molecules from scratch from an inorganic source. Talking more specifically about like carbon here. Okay, so from typically carbon dioxide. The chemo autotrophs, which are often the chemolithoautotrophs, are doing this stuff using like stuff deep down in the ocean and using, the energy from the heat of the vents and all that stuff. So, but we're really more concerned with the, photo autotrophs, right? Those were I learned later on in the chapter 24, are primary producers. So those are what they're important for. Anything hetero, or whether it's the chemoheterotrophs or whatever, the heterotrophs in general are going to be using or the organic compounds from other organisms somehow, whether they're alive or dead. If they're alive, they're typical heterotrophs like you and I. Saprobes are going to be feeding off of the dead stuff. So, but they're still heterotrophs. So keep that in mind as well. So, but all of those organic compounds come from other organisms previously. So that's what's going on with that group. So that's how we would divide them up, just be able to compare and contrast those, knowing where each one will get their energy from and where each one will get their carbon from. Whether, more importantly, I don't mean like specific, like this one from a rock or this one from, I mean organic versus inorganic sources. Okay, and that's kind of how I would look at CHOMPS in general, like where things are getting their CHOMPS from. Think of them as being, okay, well, this could be organic, this could be inorganic. Some bacteria get their S from, the rocks or something. It just general concepts there, because I'm not going to get into details on that. Right. All right, temperature preferences. This was just going into, we talked about the psychrophiles, the psychrotrophs, the mesophiles. I can't remember. I feel really bad because I know that question about the temperature is going to be on the test the mesophiles. The meso meaning middle. So that's the important word there. And then we have the thermophiles and the extreme thermophiles. So the psychro stuff deals with the cold. The thermo stuff deals with the hot. The meso is the stuff in the middle. We have the psychrophiles that have to live in the cold. They are adapted for the cold specifically. And then we have the psychrotrophs that can live in the cold, but they would prefer to be more in the meso territory. Okay. So that's the temperature preferences. Really, that's how you break it down. Most of another good thing to know is the meso zone is kind of where we're at. That's also where our pathogens are going to be at. So remember that, that the pathogens are going to be in that middle zone there, the meso zone, MESO. Moving on to the oxygen byproduct stuff. So this is a concept that carries out pretty well. And we bring up a lot, right, in class about autotrophs. Gosh, I'm already still on that. Aerobes and anaerobes. It comes up a lot. And now that you guys are getting a little bit better grasp on what that means, this is just touching a little bit on the detail of why they are the way that they are, essentially. So aerobes, they rely on oxygen. They have to have strict aerobes have to have oxygen. Those aerobes have to have some way to deal with toxic byproducts of oxygen whenever we are using oxygen as our final electron acceptor, which we learn about in that next chapter, in chapter 8, right? So when we're doing that, sometimes it's not a complete process and we have toxic byproducts, and it could be things like superoxide or, hydrogen peroxide or whatever it is, but they have to have some way to deal with it. And we have catalase and we have cytochrome C oxidase, or like that has to be testing for the ability to use oxygen at all. So you would have to have the ability to defend against it. But superoxide dismutase and catalase are the two big ones there. They're going to be superoxide dismutase, changing superoxide essentially into hydrogen peroxide. Then catalase essentially takes hydrogen peroxide and turns it into water and oxygen. And you don't need to know too much about the chemical details of that right now, but you do need to know that they have enzymes in place that will help them turn those toxic oxygen compounds into something that is less toxic to those cells. That is important about aerobes. Then we have the anaerobes. The anaerobes, they aren't going to be using oxygen as their final electron acceptor. So most of them aren't adapted to even deal with the presence of oxygen byproducts. Now, the strict anaerobes, things like Clostridium bacteria, they cannot tolerate any oxygen because they don't have any way of dealing with those toxic byproducts. There's not just toxic byproducts from the aerobic pathway. There's also toxic byproducts just in the air, like ozone and stuff like that. That's just there that, we don't really think about it because it's some small amounts to us, but to them, it's like they get exposed to it and they're immediately like, that's it. Like they're done. Like it's like radiation essentially, and it ruins them, their cells. So, so the anaerobes can't tolerate the oxygen. Okay, the strict anaerobes really, really can't. Then we have it kind of stuff in the middle. So the facultative anaerobes, they are technically aerobes. They use oxygen when it's available. If they don't have, so they can also deal with the toxic byproducts. They'll just go through aerobic respiration, just like you and I. But when there is no oxygen available, they can ferment things. And so that's where we have that fermentation process coming into play that we learn about in chapter 8 in a little bit more detail, but basically going through just glycolysis and using products of glycolysis as kind of final electron acceptors, and making acids and alcohols as products as well that we can use for whatever. I guess this whole unit kind of is really, fermentation, fermentation. You hear a lot about fermentation in this unit. Okay. Then we have the micro aerophiles, the ones that only need just a little tiny bit of oxygen to live. And then we have the aerotolerant anaerobes. Those are the ones that are anaerobes. They don't use oxygen at all, but they can tolerate it some. So that's kind of those different kinds of oxygen usage organisms, how we would look at those. Okay, so that's those guys. We talked about diffusion and osmosis. Diffusion being, movement of molecules or whatever it is that you're talking about. Movement of things from a high concentration to a lower one, just like if you had a bunch of ping pong balls piled up, they just want to go ahead and splay out all over the floor, right? That's the idea of diffusion. With osmosis, we were talking about osmosis specifically referring to selective permeability of the membrane. Membranes that surround our cells and bacterial cells are selectively permeable. They typically only let very small molecules through, including like small molecules like water. That's a kind of polar molecule, but it can get in and out. But larger things like solvents that might be dissolved, like salts and stuff that might be outside of the cell, they might not be able to move through because of their charges and everything, or their sizes. So basically the water will move instead to try to keep the balance in the cell and outside the cell roughly the same. So isotonic is whenever the outside and the inside concentrations are the same. Hypotonic, the outside, it would be like fresh water. So if your cells, which have some salts and stuff in them, right? We put those into fresh water. We're going to start taking in fresh water into our cells to try to make it the same concentration. So that could pop our cells that way. Or we have the hypertonic, where we have, like if I put you in just a vat of salt and your cells are going to start leaking out water to try to, hey, we're going to dilute out the salt around us so it's the same as us, what we have inside, but you just get rid of all your water because there's so much salt out there. That's how, another way how we preserve food. But, and that creates that osmotic pressure that we use. And when you do talk about high osmotic pressure for preserving food, that's what we're talking about. So that salt, like being high, that hypertonic solution outside. It's not always salt. It could be things like sugar as well. Anything that's higher on the outside compared to the inside. So yeah, so water will move across based on that. So in that case, with a hypertonic solution on the outside, and our water from our cells moving out to try to equal things out, basically, our cells will shrink up real tiny. It's called plasmolysis, or if you want spelling wise, plasmolysis. Yeah, so that's basically what can happen. It can kill your cells as well. Some cells have found ways to adapt to situations like that, like halophiles. Some extreme halophiles have found ways to have to live in salty environments to survive like that. They have a way to incorporate salt within their cells, so if they can live in, isotonic to them. To us, it wouldn't be. It would still be hypertonic, but to them, it's isotonic because they take enough salt in there. They've adapted to that. So if we put them into our environment, it's so hypotonic to their cells at that point that they would die. So that's how, well adapted they are to the salt. Moving on to the growth curve stuff. This one's a pretty simple one, I feel like. We have exponential growth of the cells based on binary fission. That's how the cells are going to divide. 1 becomes 2, 2 becomes 4, 4 becomes 8, 8, 16, 16, whatever, all the way up. So that's an exponential growth curve, okay? We will start with the lag phase. That's the first part, where everything's just kind of getting settled, getting used to the environment, getting everything together to see if we have enough ingredients in this environment, just adapting in general. So a little bit of a pause phase, okay? So nothing's really happening, and you wouldn't see, if you plated at that point, you wouldn't see any growth of the colonies necessarily because those bacteria haven't noticeably grown any. Then we have the exponential growth phase, or the log phase. That log phase, logarithmic phase, we have, logarithmic exponential growth. I don't know how else to describe. That's where everybody's happy. We're growing at our maximum. We couldn't be happier. And it's just, double this, double this, double this, and double this, all the way going through until we get to a point where we can just sustain the population. We're getting to that tipping point. And when we get to that tipping point, we won't be able to divide anymore. because there's not enough resources left, but we haven't started dying yet. So we'll have another one of those like plateaus there, and then we'll have the death that's exponential. Once we have really run out of our nutrients, the cells start to die, toxins are being released by the cells when they die, and everything like that. So that's exponential death after that. So that's essentially how that growth curve works and why it works the way it does. Then we have these symbiotic and the non-symbiotic relationships. Yeah, so the symbiotic is where things are going to be reliant, like you have to live in these situations. Whereas a non-symbiotic, it's non-reliant, but these relationships do happen sometimes, right? So the big ones are going to be the ones in the symbiotic ones. Those are the ones I want you guys to be the most familiar with. And those include the commensals. So a commensal relationship has to deal with the commensal organism, and we'll have one organism essentially benefiting and the other one nothing. Like one's benefiting, one's not getting harmed or anything. It's just chilling, right? So that's commensal. Then we have mutualistic. That's where both organisms are benefiting from one another. So that's mutualistic, so mutual benefits. And then parasites. So parasites they have to have a host to survive. That's part of what a symbiosis means. But basically they're going to be causing damage to their host, and that can present as disease within the host, and it often does. So causing damage to the host while they are benefiting from that. So that's the idea with that. Again, with the non-symbiotic relationships that are out there, I'm not too concerned with you guys knowing as much about those, but do know that a non-symbiotic relationship is going to be, it doesn't have to happen. They are things that happen in nature, but they do not rely on that for their existence. That's what I remember about those ones. Yeah, I think I'm okay with that chapter 7 stuff. But any questions from you guys on chapter 7, any of that stuff? Okay, cool. Chapter 8, we'll get into metabolism. I know chapter 8 and chapter 9 are the chapters. I'm aware those are the 2 chapters that people care about. This is the harder stuff. So this is breaking down stuff that you might have touched on a little bit in some of your other classes, but really got in deep on some of these chapters. So when you guys have questions, let me know. We talked about catabolism and anabolism. First of all, metabolism in general is all of the chemical reactions that occur within the cell, all of them. Then we talked about breaking that up into catabolism and anabolism. Remember, catabolism, because cats are tearing things apart, catabolism has to deal with tearing things apart, breaking bonds, and it will release energy when that happens. So when we break a bond and energy is released, we can save that energy in some other form to use to put things together in anabolism. So they kind of circle back and forth on one another a lot of times and are related a lot of times and are dependent. So, but we sometimes have to feed more into it as it gets used up, and that's why we need to eat and all of that, right? So oil rig is that term that, again, I always forget about it, like when I'm lecturing, because it's not how I learned it. I'm really trying to remember this one, but oil rig, when I see it written out, it's fine. Anyways, oxidation is loss. So that's O, oxidation, I is L, loss. So that's loss. Oxidation is loss of electrons. And RIG is reduction is gain. Reduction is gain of electrons. And I do want you guys to remember, whenever we are talking about aerobic respiration and how those electrons that serve as the means to allow us to make energy in the end, in the form of ATP, those electrons that are going to feed that whole process come along with hydrogens. At first, it's hydrogens with the electrons attached still. And that's where we have NADH or FADH2 that are going into the electron transport system. So those are electron carriers. So they're having hydrogens with electrons associated with them. It's whenever those literal electrons are going to enter into the electron transport chain with those cytochromes all lined up. And as we do that, when those electrons came off, now we have H pluses hanging around because they don't have electrons associated with them anymore. NAD flies off does its own thing. FAD flies off and does its own thing. The electrons are going to go through the chain, and those protons are going to be left over. Protons are H+. That's just hydrogen without any electrons, because it lost its electrons in the chain, to the chain. Anyways, then we pump those guys outside of the membrane, right? I know we haven't gotten there quite yet in the whole step, but it is important to remember that, because those hydrogens are how we're essentially equating our electrons. Electrons themselves aren't just going to be floating around being useful to us. We have to have some way for us to control them, or our cells to control them. And the way that we've done that is through the use of hydrogen atoms. So that's how that's accomplished. So remember, when we talk about oil rig, when we talk about the 6 classes of the enzymes, and one of them being oxidoreductase, that, this oxidoreductase, that's part of oil rig. And that has to deal with, sorry, I just got an email, a work email. And that has to deal with moving electrons around. And in our case, always think of it as moving hydrogens around, okay, for this purpose. Moving on to the enzymes, right? So the structure is very important as far as the function goes. The structure of the whole enzyme and how the substrate, which is the part that the enzyme is going to act on, how they fit together. That's important with the structure itself and the interaction of any of the molecules that are in that little pocket where the substrate would go. Because polar things might be attracted to polar things, or hydrophobic reactions might like to be, snugly fitted in there away from the polar environment. Whatever it is that drives those two things coming together, it's really important, because that's how that enzyme is going to function. That whenever it is saying, hey, I'm a C, and you come in and complete me to make the O, now I can that makes my bonds really happy. So I can scrunch down and be like this. And now that shape that has changed has allowed us to go do this other thing, or make me change you to this. And now you're floating around, and whatever it is that's going on with the enzyme, a lot of times has to deal with chemical changes, chemical interactions that result in structural changes of these proteins and other molecules they're associating with. Important to remember that. Big picture stuff. Actually, not technically, I guess, because it's chemistry, so that's little picture stuff, but concept-wise. Okay, so we talked about holoenzymes as well. We had holoenzyme being the whole functional unit. The enzyme portion that's doing the actual work is the apoenzyme, and cofactors are things that are going to come and help it do its job. They aren't part of the substrate or anything. Sometimes they can be changed. The cofactors can be changed as a result of it, and they could be used up, but they don't have to necessarily. So that's the idea with that. Coenzymes are the ones that are organic molecule cofactors. So that includes things like NAD and FAD. They'll associate with their enzymes as coenzymes, and as a result, they will gain their little electrons, so they can carry those onto the electron transport system. Okay, so we had our 6 classes, and I didn't make a list, guys. I should have done it. Let me get a paper. I probably don't have anything to write with over here, do I? Looks like I don't. I want to be able to write it down so I can actually see what I'm telling you, because I'm going to forget. Otherwise, I'm just keep saying oxidoreductase. Oh yeah, the 5th one, oxidoreductase. So let me be sure I can actually write it down. You guys know how scatterbrained I am otherwise. I'll get a pen real quick. Thanks, bro. Jeez, man. Like finding a freaking needle in a haystack. Okay, so apologize for the time. So the first one, let's try to remember. Oxidoreductases We kind of touched on that a little bit with the oil rig. That was, having to deal with the exchange of electrons. And when that exchange dealing with electrons is associated with hydrogens, we have other enzymes that we would call dehydrogenases that fall under that category. Okay, so we've covered those. Hydrogenase. Okay, and I don't remember the order on in the lecture slide, so you're just gonna have to bear with me on these. We had liases. I know that was a thing. Liases, they do the double bonds. Ligases, which we can't get away from DNA ligase, right? So when we brought up enzymes back here in this whole metabolism section, we had no idea how many times we're going to talk about a ligase. But ligase is essentially what you will do with a ligase. You will be putting things together. Thank you. Putting things together, removing water. Yes, removing water, because we have the hydrolysis exactly that we put in water when we're breaking things apart. That's the opposite of the ligases. You have to put energy into a ligase because that's the only one on here where we really talked about like building bonds. Thank you. Isomerases. Yeah, that's why I never remember isomerases. They're just changing like the orientation of the molecule. Who cares? So isomerases change basically the orientation of the molecule. Ligase, we just mentioned that we're going to be putting bonds together, taking out water in the meantime, and putting in energy to do that. Hydrolyses are the opposite of that. We're going to be adding in water. I always try to mess this part up. Adding in water while we take bonds apart. We don't have to put energy in because remember, whenever we are taking bonds apart in catabolism, which is what that will be in hydrolysis, that is actually creating energy. So that doesn't need energy to happen. That wants to happen on its own, which relates back to the second law of thermodynamics. I don't know if you guys know those, but that's why things happen that way. It has to deal with that. But yeah, that's it. Yes, okay. Transferases. Oh yeah. I feel like those kind of speak for themselves, but they're essentially going to be moving a thing from one molecule to another molecule, just transferring a thing. That could be as simple as a phosphate group. Okay. All right, so cool. I had gone through all these whenever I made all of this. I was like, yeah, here's the 6. I remember these, and now it's like, so yeah, okay. So competitive and non-competitive inhibition, I want to talk on that too. That was one of my, I feel like an important concept just to understand in general on how you're controlling your enzymes. Competitive inhibitors are going to sort of mimic the substrate that normally binds in there. The enzyme acts on the substrate. Typically, the competitive inhibitor comes in, binds in that same spot where the substrate would bind. The enzyme can't act on that, and it often gets stuck like that, and that's the end of the reaction. The enzyme is done working. So that's competitive because it binds in that same spot as the substrate would. The non-competitive inhibitors, they are usually products of the reaction. So if you are, making some sugar as a product to store in your cells or whatever it is, you're actually making it. And your enzyme that's involved is working on the substrate that allows you to do that. So enzymes work on the substrate, We're making more and more of the product. Eventually, enough product comes from this reaction that it will bind onto another area on the protein that is not the actual binding site or active site where the substrate would be interacting. It will bind somewhere else on that enzyme and usually cause a structural change in the enzyme. Now the substrate can't even associate with that enzyme anymore due to that structural change. It's non-competitive because it's not binding in the actual active site itself. Cool. Not competing for the active site. So yeah, so the biggie on this whole chapter, even though understanding enzymes and what they are in general is an important one in here, the aerobic respiration, right? That was the biggie here. Aerobic respiration, of course, dealing with making your energy and using oxygen as your final electron acceptor in the whole process. The anaerobic version of this, we have using anything else, not oxygen, using something else like a nitrate or nitrite, whatever it is, that organism uses something else, but it's a similar process that they'll go through. Then we also have fermentation, and where we have typically in aerobic respiration and full-blown anaerobic respiration, a glycolysis step, a Krebs cycle or citric acid cycle, and the electron transport chain. And most of your energy is being made in the electron transport chain, by the way. Your fermentation really only goes through glycolysis. And where normally our electrons in this whole process will be dealt with or recycled or used up, taken care of. So they can't just float around forever on their own. They get reabsorbed into nature in a way by oxygen or not oxygen at the end in the electron transport system. In fermentation, they don't go through that at all. So they have to do that with organic molecules that are made at the end of glycolysis, like pyruvic acid, and which is the main outcome of glycolysis. And so whenever they get that, and they'll use their electrons and mix and match whatever they want to do with that molecule, pyruvic acid, depending on the organism, their byproducts are going to be alcohol, like drinking alcohol, typically ethanol, or acids of some kind. Like lactic acid is a very common byproduct. And that's what we get when we're talking about things like when you ferment pickles or sauerkraut and stuff. That's why it's sour the way that it is. Okay. Next, well, not next, but I mean, like that's the compare and contrast of the basic rundown of those. And if you understand the steps of aerobic respiration, which I highly recommend that you do, then you will understand anaerobic and fermentation, because the little differences there, that little difference for anaerobic, not oxygen as the final electron acceptor, that's the difference. And then fermentation being stopping at glycolysis and using that product for making alcohol or acids byproducts when they're making energy. They don't make very much either. Only about 2 ATP overall comes from glycolysis. And I don't need you to know those numbers, by the way. I don't need you to know exactly how many ATP is made in glycolysis or in the Krebs cycle or in ETS. What I do want you to know is which one makes the most. And you should be able to tell me that it is the electron transport system. It makes by far the most of the ATP. Okay. Remember, ATP is the molecule that is our goal here when we're creating energy. It's going to store up those energy from breaking those bonds earlier on in the catabolic process, right? The first step in all this is glycolysis. We start out with a sugar, typically in this case where we were talking about glucose. Okay. We will start out with glucose. It is a 6-carbon molecule. We have some changes that go on in there, chemical changes that you don't need to care about. You will make eventually your first 3-carbon molecule in double, which will be 2, each from 1 6-carbon sugar, you will get 2 3-carbon glyceraldehyde 3 phosphates. Okay, so there's that. So we made that, cool. And then that's going to be processed down through a bunch of steps into our main product of glycolysis, which is pyruvic acid. So from 1 6-carbon glucose, we get 2 glyceraldehyde 3 phosphates. That each of those will get turned into 1 pyruvic acid. So we get 1 carbon or 1 6-carbon sugar into 2 3-carbon pyruvic acids. Okay, so what does it matter? What do you care? What is the whole point of this? We're prepping our sugar to go through the Krebs cycle, which is important, and the electron transport chain, which is extremely important, so that we can make energy. This is like a preparatory step more than anything. We do get some ATP formed here. If we didn't get ATP formed in glycolysis, fermentation wouldn't work. So we have that going on in our favor. We do make some energy here. We also make electron carriers like NADH. Now, NAD gets turned into H, it gains that electron. So if it is gaining the electron, it will be called reduced. So it is being reduced into NADH, okay? So yeah, it gained its little electron, and that's how we're going to carry our electrons around in our electron carriers. So we've done a little bit of that in glycolysis. We've prepped everything and gotten everything ready to make our pyruvic acids, okay? Each pyruvic acid will be turned into a molecule called acetyl-CoA. Don't care about how that happens, but just know that it does. Acetyl-CoA, it's the coenzyme A is what it stands for. Acetyl coenzyme A will go through the Krebs cycle. You have to be that before you can go through the Krebs cycle. The first step is to get it turned into citric acid. You don't need to know that, but that's why it's called the citric acid cycle as well. So we're going through the cycle, and we're really just like knocking off a couple bonds, and we're changing up shapes again and doing some of the stuff like we did in glycolysis, and some of the stuff where we're actually removing carbon molecules off of this stuff, okay? And shifting around our hydrogens and taking them off and putting this there and put whatever. So we're making in the process some ATP. We are making electron carriers, which are extremely important. So a lot more electron carriers are made in the Krebs cycle. That's the importance of the Krebs cycle. That's the main take-home message of a Krebs cycle, is that you're going to be making those electron carrier molecules. We're reducing them, so they're carrying now our electrons as we go through that whole cycle. That's the whole point of it. And then we also, in this cycle, is where we start shooting off our carbons in the form of carbon dioxide. So Krebs cycle is where you exhale, why you need to exhale carbon dioxide. It's that's the reason. So we have gotten, breaking down our sugar into 3 carbon molecules. We've changed them into acetyl-CoA and shoved them into the Krebs cycle. And now we've taken the energy out of their bonds and put that energy essentially still stored in those electrons to carry around in electron carriers. And now we're going to go shuttle those electron carriers over to the electron transport system and pop those electrons into the system as a result. So remember, we had NADH as one of the electron carriers, right? So that's it carrying the electron. NADH gives its electron, the actual electron, to the transport chain molecules. Those molecules are called cytochromes. That's all you need to know about them. And they're lined up, and they just go and excite the electron will excite each one as it goes down. But you know, NAD plus, NAD goes off on its own. We don't need it anymore. It's done. It did its business. Now it can go become NADH again, going through Krebs or whatever. So it's done. So we have NAD done. We have electron gone. So what you're left with is H plus, which is hydrogen with no electron, because it's gone. It went in the electron transport chain. And hydrogens typically only have 1 electron. So now it's H plus. We call those protons. So they essentially are, right? Because they don't have any electrons. We don't care about their neutrons. So all they really are is a proton. So as the electrons are going through those molecules and exciting those cytochromes on there, those protons are getting pumped out of the membrane on the other side of that membrane. And we're getting this massive amount of protons and creating the proton motive force. All those protons are going to build up out there. They can't come back inside the cell anyway, because they have this positive charge, and that goes against all that negative on the cell, which we keep talking about with the stains, right, with those phospholipids. So they can't come back into the cell just on their own. We build those up. We've got a negative charge associated with the cell, especially while we're chucking those electrons down there. So what they have to do is they have to come through this molecule at the end of all of this called ATP synthase. And exactly like it sounds like, you're going to synthesize ATP because those protons are moving through that protein. It's like a little tunnel, basically, what gets associated with that enzyme. It's an enzyme as well, ATP synthase. Proton will come through, and it changes the shape of the ATP synthase, allowing it to put a phosphate group onto ADP to make ATP. Okay, So that's how that whole system works. So now we have, we shuttled all those protons out. We've brought them back in slowly. So that's nice. So at the end of all of this, we have an electron that's made it to the end of the chain. And we have these protons that are building up and back inside of the cell because we're bringing them through to make ATP. Basically, what do we do with all of that? So the idea is that, get rid of the protons, get rid of the electrons. So we take oxygen and split it up and take 2 protons, 2 electrons, and 1 oxygen, and we make water. So all that is the byproduct of the electron transport system to help get rid of those stray electrons and stray protons to equal all of that out. So that's how that's all maintained and done from start to finish. There's another enzyme that helps with that process, making the water from the byproducts and the oxygen. The enzyme that does that is called cytochrome C oxidase. And like I've mentioned previously, if an organism has that, they are going to be in some way able to use oxygen at some point in their electron transport chain, whether they're facultative or strict aerobes. So you can test for that and see if something has that. It's pretty simple. You just swipe them on these like cards or put a drop on them and it turns a color. And if they have it or not, it turns a color. So it's a pretty neat little test. You guys will be doing it in the unknown stuff. But anyways, that cytochrome C oxidase, obviously associated with cytochrome C, which happens to be the last of all of those cytochromes that we had lined up, associates with that to get rid of those byproducts. So that's what's going on there. So we added in that part of that oxygen, the protons and your electrons to make the water. The oxygen is why you breathe in oxygen. So we breathe in oxygen in order to help eliminate that final electron in the whole transport chain. We breathe in oxygen for that, and we breathe out carbon dioxide because of the breaking down of the carbon molecules in the Krebs cycle. That's why. Okay. Okay. So that was aerobic respiration, everybody. I feel like I kind of already covered fermentation. Again, anaerobic respiration, where we have that oxygen coming in, there's a different process involved for anaerobes that involves something other than oxygen. It's a similar process, but not oxygen, other things. And fermentation just ends at glycolysis and using up those products to make alcohol or acid as a byproduct. Any questions on that part, on the aerobic respiration before we move on? Okay. I don't know if anybody, I'm assuming nobody's actually talking. I mean, I know that we have the chat, I have the chat open. I'm sure you guys can see that. I have the chat open so you guys can chat with me. If anybody's trying to talk to me, I can't hear it. So I'm assuming nobody is. I'm trying to. I feel like it should work, but it works when I play games. I'm at home on my gaming PC right now. So I don't know. I hope it is working okay. So photosynthesis. Basically, that is like a pre-step. So we are using the energy that is within bonds of organic molecules that other organisms made. We don't make our own. We have to break up other ones in order to get the energy and the building blocks for our own stuff. That's what we talked about in chapter 7, right? With autotrophs versus heterotrophs. So photosynthesis is for those photoautotrophs. Photosynthesis, we use energy from the sun, and it excites it excites pigments that are a lot like the cytochromes. Okay, so we can essentially have electrons getting excited or molecules getting excited involved there, and using that to create a proton gradient, and then using that to drive ATP production. Now, in that first step, the light reaction, where we're getting light and interacting with the pigments and then resulting in some ATP, right? We don't get very much ATP, but we get enough. We get enough to start into the Calvin cycle. And what's the Calvin cycle? The Calvin cycle is the dark reactions in plants or other photosynthetic organisms. So the light dependent ones, where we're getting light, turning it into a little bit of energy. And that's all that's going on. The light independent or the dark reactions, the Calvin cycle, we are literally taking carbon dioxide from the air and using the energy from the light portion to stick carbon molecules together to make glucose. That's not it for plants. In that whole process, in that dark part, we are taking up carbon dioxide and producing oxygen. So the amount of carbon dioxide being taken in and the amount of oxygen being given off is enough that whenever plants have now their glucose, guess what plants do with their glucose that they just made? Well, they need energy too. And plants, they don't just have chloroplasts. They also have mitochondria. Now I'm talking about plants because it's easier to separate it there. But anything photosynthetic will do something similar to this. Even if they don't have separate mitochondria, this will be going on the inner membrane of bacterial cells. But anyways, they will just go into aerobic respiration, and that's how they get the rest of their energy. Plants, we're talking about plants, they obviously don't need quite as much energy as you and I do. They're not running around, they're not going in marathons and all that. But yeah, so that's the idea. So yeah, they still do aerobic respiration, which you need oxygen for. But the thing is with them is that they're giving off enough oxygen, so much more that they still give off enough for us to breathe in our atmosphere. So that's pretty successful, right? That's a good thing. So that is that on metabolism. That is that on aerobic respiration, photosynthesis, enzymes. Chapter 9 had to do with genetics. First thing we'll go over is a phenotype versus a genotype. The genes are what encode, your traits. So a gene, basically, you could have a gene from your mom and a gene from your dad, and obviously they could be different from one another. We know they could be, right? You would still be carrying those genes, but you'll only see one of them expressed, not both at the same time, unless you have like heterochromia or something where you have like different colored eyes and stuff like that. And that's a weird case, rare case, and you don't see it a lot, and it isn't related to the same thing. So basically, if you have two genes for something and they're different, one of them will be expressed, typically, not both. So what is expressed, what's actually expressed, what we see, that's the phenotype. Okay, so genes is what's in your genes, your DNA, what it says, and then phenotype is what you're actually seeing produced. Okay, DNA versus RNA. DNA is deoxyribonucleic acid, and RNA is ribonucleic acid. So there's two major differences in DNA and RNA. Well, I mean, typically RNA is single strand. Is there a difference between genome and genotype? Yes. So the difference between genome and genotype is the genome is referring to the entire genetic makeup of an organism. That will include that organism's chromosomes, like all 46 of ours, the 23 pairs, as well as any other DNA associated with the cell. That could be, for us, the DNA in our mitochondria, because remember, they have their own DNA that's different from ours. And for bacteria, it could include like extra plasmids or whatever else. So that's the genome, that's the whole genome for the whole thing. And that is just referring to the whole makeup of it. The genotype is talking about the exact genetic makeup. So what type of gene you have. That would be breaking it down, the whole genome, referring to, okay, these are all the genes that this organism could have in here. Whereas the genotype would be saying, okay, this gene has for brown hair from the mother, and this gene has for blonde hair from the father, that sort of thing, breaking it down into actual like what the genes would actually code for. So that's going to be the genotype. I hope that answered your question. Okay, so DNA, vector DNA, and RNA. RNA tends to be single-stranded, typically when it's functioning in the cell for us, and DNA is double-stranded. We know the double-stranded helix, right? That looks like a ladder twisted. And so that's one little difference. the big differences come from the sugar. So both of these are consisting of a nucleotide backbone. A nucleotide consists of sugar, a phosphate, and a nitrogenous base, right? For DNA, the sugar is always deoxyribose. For RNA, the sugar is always ribose. So it's different sugar. The only difference is there's an oxygen removed for DNA. That's in the name. So that's the only difference. It really is. So that sugar is that difference. Okay, so that's how the sugars are different. The other big difference with DNA and RNA, we know that DNA has A, T, C, G. Those are referring to the nitrogenous bases. So the 4 possible nitrogenous bases that make up our genetic code are being associated with the DNA. Now, when we go into that DNA blueprint to make a copy, a working copy to take out to make a protein in the form of mRNA or messenger RNA, when we go in to do that, and we see a C. So on the RNA, we would put a G. We see a G, we would put a C. Again, a G, we would put a C. We see an A. In the RNA version, we would put a U, not a T. RNA does not have T. It uses uracil instead of thymine. So instead of adenine, guanine, cytosine, thymine that we see in DNA, we have adenine, cytosine, guanine, and uracil in RNA. And it just has to do with how those molecules are going to interact with what they have to interact with, like ribosomes and things like that. So that's why that difference exists. So T replaced with a U. So those are the big differences in DNA and RNA. And obviously DNA is going to be what codes for your actual genome, and the RNA is going to be in the form of the biggies, tRNA, the transfer RNA that carries your amino acids during translation, rRNA that makes up your ribosomes, and messenger RNA. Those are the working copies of the genes that we're going to be using as a template to make our proteins, right? So those are our RNAs, and we have considerably more RNA in our cell than we do DNA, because we only have one copy of our DNA. You only have one of your, that chromosome or whatever it is. So you make copies out of that, and you can have several copies of the same thing going on to make more, production greater, right? So that's the idea there. That's why we have so much more RNA, and also ribosomes and transfer RNA and stuff. So, okay, the next thing was DNA replication. Anytime that you want to, replicate your DNA, you're going to have to unwind that helix. So first thing, we're going to unwind the helix. And so we have, the A's with the T's and the C's with the G's. We split it apart. So we've unwound, we split it apart, and we always add nucleotides from the start and end will always be 5' towards the other side, so 3'. So the other side will always be 5' to 3'. So we'll be going opposite directions. 5', 5', going in. Let me draw it on here because I'm talking about it like an idiot, and I feel like I would be easier because you guys can probably not see me that well. My monitor is huge, so to me, I look giant, but so you might start here. Oh, am I clicking? I don't know. And then go out in this direction. If you started out here, this is the 5'. So 5' is always the start. It looks like the S. So that's how you can remember. Then the other strand, when you're copying whatever's going on here, will start on the opposite side and go the other direction, because that's just how it works. So this is 5', that's the start on that one. So they're anti-parallel. That's what anti-parallel means. 5' going this way, 5' going that way. Okay. So that you know what that means, anti-parallel like that. And 5' and 3' are referring to specific carbons in the sugar structure. That's all they mean. It's just numbered. They numbered the carbons, and it's not completely arbitrarily. There's a chemical reason for why they did it the order that they did. but you don't have to worry about that. You just have to know that the 5' end is the start of it. You can always have to start there if you're making it and go in the other direction. Okay, so if this was the 5' to 3', and we're opening up our, helix over here on this end, right? And we're going to come in and start making copies. And this side, as we're opening up, could just keep pushing in and pushing in and pushing in and pushing in from its little 5' down here, going in this way, adding on at the 3' end, starting from the 5' end. That would be the leading strand. This top one here, as this opens up this way, so this is where that ladder is all tightly wound, we're unwinding and opening it up further and further towards this direction. So we're getting little parts opened up in spaces. So we can come in and have a start here from our 5' and go a little bit of a way and meet up with the other one where we went a little bit of a way there. And as it had opened up that whole way, we could only make it in fragments as it opened up. Okay, so one goes directly, but we can only add in 5' to 3' direction. We can't go 3' to 5'. So both strands can't go into that opening. So what we do is build it from in pieces. And those pieces that we make, 5' to 3', as it opens up, those pieces are called Okazaki fragments. Okay. And I'm sure you guys remember at this point. And remember also that whenever we are adding nucleotides for DNA, you have to have a primer there. You can't just stick them onto nothing, into air. So they have to have a primer there to add, to link onto, and that's typically an RNA molecule. And then another enzyme will go in and break that apart, and then we'll fill it back in with DNA. So primers are needed for that, natural primers in your body. So we have that. So whatever, we did our primers, we replaced them with DNA. Now we still have our fragments. I'm sure you guys at this point could tell me what enzyme is going to come in and seal those fragments together. If you can't tell me what enzyme it is by now, because I feel like I've said it 1,000 times. I'm going to tell you anyways, but I feel like you should probably know this one, that the enzyme that's going to come in and seal DNA pieces together, like it always does in any DNA when it seals DNA together, that is DNA ligase. Okay, so DNA ligase is going to come in. Remember, ligases are going to link things together. They're going to require energy to do that. But basically, that's a DNA ligase. So it comes in and just seals everything up, joins the bonds together. So that move done by that enzyme, that's an anabolic reaction right there, because we made bonds. Cool. So that's anti-parallel, right? So if one's going 5' to 3', the other one's going the opposite direction. So whenever we are done making this, we had one strand that was the template strand, and the bottom strand was also another template strand. And we had 2 new strands that were made off of each of those, right? So we have an old strand with a new strand, and then an old strand with a new strand. That is semi-conservative. We conserved one strand, made a new strand. So semi-conservative. That's what that term means there. Okay, moving on. Now we can go transcription to translation. Transcription to translation. And then we're going to compare prokaryotes versus eukaryotes. Cool. I put that little note in there because otherwise we wouldn't have thought about it. Transcription is taking DNA, blueprint of DNA, and literally just making a working copy. That's what I like to refer to it as, because I feel like it's the best way to understand it. The DNA blueprint, making a working copy into RNA in the form of messenger RNA. That's the process of that. So literally the RNA polymerase will come and bind onto the promoter area on the DNA. It knows where that is. And it will just start adding on nucleotides until it's done. That's that. It's pretty straightforward, is what I'm trying to say. So at least that part is pretty easy. And you make your RNA, and that's that. And then we're going to take that to a ribosome. And then the ribosome in translation So we had to write down our working copy first. Now I can take that working copy out to the ribosomes. The ribosomes are going to translate it into protein, which is like another language. The others were nucleotides, same language. We're just writing them down, making a copy. Now we're going to translate it into a whole other language, which is proteins, amino acid sequences. So we take that messenger RNA out to the ribosome. And what happens there is the ribosome will bind onto it. And on the messenger RNA, we start with the start codon. And that is a 3 nucleotide sequence, usually AUG on the messenger RNA. You don't have to know that, but it just is that. Where you will have the transfer RNA, which carries an amino acid on its little end. It will come in, and the one that matches AUG, which in turn would be, so A would be T, so it would be U, U, A, C. Yes. Okay, I had to think. I should have just wrote it down. I don't know why I'm trying to think it through. I'm really bad with seeing things in my mind. But yeah, so if you had AUG, it will be UAC on the anti-codon. So they're always in 3s. In DNA, that would have been the triplet code. In RNA, it's the codons in 3s. And then the transfer RNA has the anti-codon that matches it. Okay, so after we've matched up that complementary sequence from the mRNA to its appropriate transfer RNA, it has its associated amino acid attached to the top of it. And the next one can come into the next pocket of the ribosome and match the next codon, whatever. I don't know what that is. The next codon and whatever, I don't know, it carries another amino acid, right? Those 2 amino acids that we have now, those will get linked together through a peptide bond. Remember, we learned about those in chapter 2. I'm sure you remember it. Peptide bonds that'll link them together. And it'll shift the ribosome over, kick off that first transfer RNA. We don't need it anymore. We don't need it anymore. It'll go back and collect a new amino acid to do its thing all over again somewhere else. So yeah, so we'll shift it down and then we'll add in the next one and the next codon and then shift it down and so on and so forth. So that's how translation is going to work based on your codons. Now there are 20 amino acids and way more codon versions than that, whether it's CCZ or AAU or whatever. They all code for something. Some of them, some of the amino acids are coded for by more than one codon. And that has to do with that wobble effect. So they can code for more than one codon because that helps us kind of deal with any mutations that might arise and account for any changes in the amino acid sequence that could cause a structural change in our protein, trying to prevent that. So that's what that's about. It's useful. I think that's that on. So yeah, so we talked about triplets. There are 3 nucleotide sequences in the DNA. The corresponding 3 nucleotides in the messenger RNA are the codons. The 3 corresponding nucleotides in the transfer RNA that matches those codons, the opposite of it, are the anticodons. And then the transfer RNA that matched carries the amino acid associated with that codon on the messenger RNA. And that's how we get proteins. Just adding on those amino acids and peptide bonds, eventually have this giant, big old protein. Okay, so we're going to talk about the difference between prokaryotes and eukaryotes. Prokaryotes, keep in mind, prokaryotes like bacteria, their genome, their chromosome is just hanging out in their cells. They don't have any membranes around it. It's not contained. So all of this is just happening all in the cytoplasm because that's all they are on the inside is cytoplasm. They don't have anything else in there. Well, they have ribosomes and stuff, but nothing like they don't have, mitochondria or endoplasmic reticulum or any of that. They don't have that. So that can just go on in the cytoplasm. The good thing about that for prokaryotes is that because we can make messenger RNA from the DNA right there, make our blueprint into our working copy right there in the cytoplasm, ribosomes can come and attach onto that working copy and start making protein before it's even done being transcribed. Like while that's happening, it can do it. So that's the good thing for prokaryotes. We cannot do that. Our DNA to RNA happens in our nucleus, and then RNA to protein happens outside of the nucleus, typically in the endoplasmic reticulum. Okay, so that's the difference between those. Operons. We had inducible versus repressible operons. I do believe that the inducible example that it gave us was the lac operon, and the repressible example that it gave us was arginine. I think it was arginine production. It doesn't really matter. But basically, inducible operons are ones where you don't want to be, making an enzyme or whatever that's related to breaking something down if it's not there to be broken down, right? So that's the idea. So in bacteria, they can string genes together that code for the same executable process. So if you need to break down lactose, but you need 3 different enzymes that are completely different from one another in all those steps, but you need all 3 of them, they have all those genes linked together in one, all back to back. And it's all controlled at their very beginning of all of this. So their promoter region and operator region and all of that. So the promoter, again, it's just the part of the DNA that the RNA polymerase attaches onto to make the messenger RNA. That's all that is. The operator region for these operons is the area where the repressor is going to associate. The repressor in the example that was given in the slides looked to me like a Cylon spaceship, which probably means nothing to any of you, but it's Battlestar Galactica. And back in the day, man. But that's what it looked like. So it was like a horseshoe almost sort of shape. So horseshoe shape that came and bound onto the DNA so that we can't do our RNA polymerase thing. We can't make messenger RNA. Okay, so that happens. And we can then come in and introduce lactose, and it will bind onto that repressor protein and change its shape because of the chemical interactions. And then the repressor comes off, and we can start making those enzymes because we've got lactose now. So that got triggered, the repressor got triggered that way. And eventually you're going to use up the lactose and it's going to get broken down and everything, and or more repressor is made and it's not being blocked anymore. And you can go back to repressing all of that when you don't need your proteins anymore. So that's inducible. We had to induce it. Repressible operons, the way that they work, they're always on. And what they're going to be doing is making like something. So in this case, it was like making arginine. I can never remember it. And I have to keep reminding myself, looking back in like my memories of what I think was arginine. I'm going to write it down so I don't forget it again. So in our repressible operon, we are making all of those enzymes. Again, it's just a whole line, a whole slew of enzymes in this line. Just always making enzymes associated with arginine, let's say. Okay. So our promoter is happy. We have our RNA polymerase at the promoter. It's happy to go down and start making the messenger RNA because nothing is blocking it on the operator. There's no repressor there. So everything's happy. We're making the messenger RNA, making the proteins associated with making arginine. Basically, if you get enough arginine going on, you would be able to, or things that are associated with its metabolites. We have arginine made, our cells have used it, things are made as a result of it, and now those can come back and associate with the repressor in a way telling the cell, hey, we have made enough of this. We don't need to keep making more arginine, associate with the repressor, and it comes and binds onto the operon, much in the same way the other one did. But we started off with making, and now we are repressing. So it is repressible. Cool. I think that's that for that. Okay. We talked about horizontal gene transfer in bacteria, which just has to do with those 3 different ways that bacteria can spread their genes, not from their parents, which would be vertical, but between one another. We had conjugation, we had transformation, and then we had transduction. So those were the three ones. Conjugation, direct contact, the bacteria come together, they make what is called a sex pilus. It's just a tunnel between them, and they can put their DNA through there. It could be just a plasmid, it could be part of their genome, it doesn't matter. That's how they exchange their DNA. The transformation one, that is picking up DNA from their environment outside of them. So the DNA just chilling, either in pieces from some dead cell or something that maybe a genetic engineer introduced to them. If a cell is able to take up that stuff around it, the DNA from outside of it, then that cell is referred to as competent. It is able to do that, so it is competent at doing that, okay? So the competent cell takes up the outside DNA, it can incorporate it into its genome, or if it's a plasmid, just have a plasmid that confers like antibiotic resistance or something. Okay, that's transformation. Transduction is when viruses carry genes to new bacteria. So we've infected one E. coli, maybe our cells start, we have infected E. coli A, making viruses as a result, building up virus particles in there. One of our virus particles takes up part of the A's genome, just randomly. Then it gets out, they burst through the cells in bacteria. Bacteriophages do. Bacteriophages go everywhere. We go to another cell, to E. coli B. We infect it, inject in what we think is our genome, but is really just a piece of the other A's genome into that bacteria, and it can incorporate it into its genome, because bacteria don't always know, so they're not always that smart. So you can transfer any gene that way. You can't control what gene that virus is going to carry around with it. But there are some ways that you can, in a way, know what gene is going to be, or you can predict what genes will be passed on by viruses. And that is when the virus is a temperate bacteriophage. Temperate bacteriophages are the ones that can go from the lytic phase. Normal viruses come infect the cell, we make our parts, and we bust out and keep doing that over and over again. They can go from that to the lysogenic phase. In the lysogenic phase, they incorporate their own genome into the bacterial genome, kind of to hide out for a while, for whatever, waiting for a trigger or whatever it is that they want. And when they are incorporated in there like that, they are called a prophage. So they are a temperate prophage in that. Prophage for them. For us, if we had something similar in people, we call that provirus. Same thing. So, right. So we know they typically, when they insert themselves in there like that, they become in the lysogenic phase, the prophage. When they do that, they usually do that in predictable spots. So they come in and they, park themselves next to the bacteria's gene for a capsule or something. And then when it decides it wants to make more bacteria, sometimes can take capsule gene with it and go to the next bacteria B, and put bacteria A's capsule gene in when it tries to become a prophage. So that is how that can happen. That's a horizontal gene transfer through transduction. Then we talked about the different kinds of mutations. I should have made a list of that one too, but right. So we had the point mutations. We had the lethal mutations. That you know what a point mutation is. It's one nucleotide getting changed, either added or subtracted or just changed. That's a point mutation. Just one of them. Lethal mutations are any time that you have a mutation that kills the cell. And then we have basically silent mutations, which was included in the next slide, but it was basically talking about the same thing. Ones where you have some kind of change, but we don't see any effect on the cell. So it's like the general breakdown. And then we had the specific breakdown, missense mutations. A missense mutation, remember we had talked about codons. Codons come in series of 3 nucleotides, right? That codes for an amino acid when it is read by the anticodon within that ribosomal process of translation. That codon, if let's say you had let me write down, I'm gonna forget AAG. Let's say you had AAG and it got turned into, by a mutation, a point mutation, it got turned into AAC. Let's say AAG coded for alanine, it's an amino acid, and now AAC, I mean, I'm trying to, alanine is, let's just go with lysine. I don't like this. Okay, sorry. I'm trying to make it complicated on myself, and I shouldn't be, because I was trying to think of different, because I was about to say phenylalanine, and I was like, wait, I don't want to make them sound the same. AAG, let's say it codes for lysine. I'm sorry if that's really loud. That seemed like it was loud. Lysine. AAG seems like it sounds, codes for lysine. AAG gets a point mutation to become AAC. And now let's say that AAC actually codes for tyrosine. I don't know. That's a different amino acid. All right. That's our example. But we only had one little change. One change led to a change in the amino acid. That is missense. It took me that long to get to it, but you get the idea. We had a change in our codon, changed the amino acid, missense. Okay. Nonsense, we have a change in the we have a change in the codon, which could be like AAG, which was for lysine. Now we have a mutation for it to become, instead of AAG, UAG. And UAG does not code for an amino acid. That makes it a stop codon. Stop codons are also known as nonsense codons because they do not code for an amino acid at all. So that is a nonsense mutation. It's nonsense. We don't get an amino acid as a result. So anytime you have a change that results in the amino acid being changed to a stop codon or not coding for an amino acid, that is a nonsense mutation. Okay. We also have silent mutations. If we had AAG and it codes for lysine and it got changed to AAA, but AAA still codes for lysine, who cares? That we did have a nucleotide change. It is a mutation, but we had the same amino acid, therefore the same exact protein shape, everything. So who cares? That's called a silent mutation. Same amino acid still made. We did have a mutation, but same amino acid. That's all that really matters. Okay, here's another example, a back mutation. A back mutation is whenever we have gone from something being like a bad mutation, a mutation that deviated from everybody else, and now we are reverting back to the original. That's pretty straightforward, right? Going back to what it originally was before we had this weird thing pop up already. Back mutation, reversion. The other one is frame shift mutation. I hope that's all of them. The frame shift mutations, basically what you'll have there, if you are reading in steps of 3, let me write this down. Oh, let me actually draw this one because this I feel like would be a good one for, I wish I could type it, but I don't know how to do that. So if I had AAA, I just try to go with the easy stuff. And then U, and then G. Now this is the mRNA, of course, that's what I'm trying to say here, but DNA, this would be TTT. Okay, so this is our mRNA version that our ribosome would be reading. And let's say in a frame shift mutation, you insert, so randomly, you just decide we're going to insert, I don't know, another A. Well, everything is still read in the form of 3s, right? So we would still have AAA, and we're good there. But now we have AUU, and then we have UGG, and so on and so forth. So everywhere after where we put this new A in, everywhere after that is going to be messed up. You see how that works? So, because you still have to read it in 3s. You can't just say, oh, this wasn't supposed to belong here and keep going, U. UUU codes for something completely different than AUU. I can assure you that. I don't know what, and I don't really care that much, but UGG would code for something completely different than GGG. So now suddenly, instead of, the AAA UUU GGG resulting protein, now we have something else that has different amino acids, completely different amino acids from that point on, and would probably be completely useless to us, at least as far as that protein was concerned. We're not trying to make whatever this mess is. We're trying to make, AAA, UUU, GGG. So that's the idea with a frame shift mutation. This will occur with any sort of mutation in like ones and twos, multiples of ones and twos. But if it's a multiple of 3, we're still going to maintain that reading frame of the 3s. So that's one aspect about that to remember. So 1 and 2 nucleotide changes, inserting or deleting, are going to have the most devastating effects. Sorry, I'm reading this real quick. Right. Yeah, so as far as the mutations themselves would go, we can have spontaneous or induced mutations. I do want to touch on that. The spontaneous mutations are just anything that comes up on their own due to just errors in cells and how they are copying their DNA and everything. It's not perfect. So sometimes we have errors that will pop in there that got missed for whatever reason. It's more common in things like bacteria and viruses. They don't have quite the extensive checkpoint program that we do. And also turns out with their high reproduction rate, they can kind of afford to lose some guys to some mutations because there's so much of an advantage to gaining, a mutation that would be helpful. There's so many of them. Expendable guys, get rid of them. So that's the idea. That, and then the induced, of course, that's going to be whenever we have something that is going to cause mutations like ultraviolet radiation or certain chemicals that can literally cause mutations. As far as mutant strains go, that's just be anything that could have change in the sequence that would differ from the normal predicted wild type sequence. I think that's it. All right, genetic engineering, chapter 10. That chapter was not quite as bad. So we had talked about DNA size. Please keep in mind when you are reading the numbers to make sure that what kilobases means, and megabases being 1,000,000 and terabases being 1,000,000,000. Be aware of that compared to a base pair, just one base pair. So if I had like 1,000 base pairs versus 2 kilobases, 2 kilobases is more than 1,000 base pairs. Can't say it enough because it seems to trip people up. I think they just read too fast and get too stressed out or whatever during it. But FYI, there's that. I said it again. Okay, so we measure DNA size based on the base pairs. So I would say, A, T, C, G, that's a base. Base pair is referring to A and its bound partner across the way, T. And G, its bound partner across the way, C, and so on and so forth. So those pairs that are together in their bound partner setup. Okay, that's DNA. Agarose gel electrophoresis. That is one where we are taking, I don't know, sorry, my little rolly pad here has like a tilt in it. And sometimes I just roll off into oblivion. Right. Agarose gel electrophoresis. Okay, so basically we're going to take a gel. It's not unlike the agar that we use, but it's just a clear, purified version of it. Put that in a nice little buffer, charge some electricity through it. We put DNA into these little wells at the top. Okay, so that's cool. So we're running this electricity through. The DNA has a negative charge because of all those phosphates, right? Those phosphates are going to cause that negative charge DNA to move towards the positive pole while we're running electricity through. So they'll do that and they'll run from that top where the wells were, where we put the stuff in, down to the bottom towards the positive. The bigger ones are going to get stuck in the top. The smaller ones are going to move faster through it. That makes sense if we're thinking about, large like basketballs running through something versus like ping pong balls, right? I just keep those stretchy nets that like hold the balls in or whatever. I don't know if that makes any sense to you, but it's like always reminds me of. But yeah, the stuff is stuck in there. It'll move through eventually. And then so when you're done, you can visualize those with special dye and look for the larger bands that would be at the top and then the smaller bands towards the bottom. And if I digest with restriction endonucleases, remember, they're going to cut in specific palindromic sequences onto DNA. And if I cut mine with ECO-R1 or HIND3 or whatever, and then I cut yours with ECO-R1 or HIND3 restriction endonuclease, my pattern will run out differently than yours will on the gel. And then we can compare and contrast those restriction fragment. There's a polymorphism. That's the word, polymorphism. Anyways, we can compare those to one another. For sure, I can't talk today. I shouldn't have woke up this morning, I guess. We'll compare the sizes so we can say whether somebody is related because they'll have similar or not, or they did or did not commit the crime or whatever. So that's a quick way to do that other than just running the PCR, which is another way to do it, just sequence it. That's really how people do it nowadays. But yeah, we can also make sure, okay, if I wanted my plasmid to be 1,000 base pairs and I'm, cutting it up and I'm going to get one that's 1,000 base pairs and then one that the insert that I don't want is 500 base pairs. Now I can separate them out on the gel. I can actually cut it out of the gel and dissolve it out and purify it and get only the plasmid, not the part that I cut out. So that's another way to look at it. Useful. Hope that answered everything for that part. PCR polymerase chain reaction. It's a lot like the DNA replication. We are going to have DNA polymerase. It has to be special because it has to withstand a lot of heat because we're going to be heating and cooling and heating and cooling the whole time. And the heating is meant to denature or separate the DNA strands. That's what denaturing means. It means like basically taking it apart, ruining the chemical bonds. Denaturing or ruining the chemical bonds of DNA with heat typically would do so to proteins as well. So we have that special DNA polymerase that is from a bacteria that lives in like the hot thermal vents in the ocean. And we've extracted its polymerase to use in this process. So it can withstand a lot more heat. Essentially, we will heat our DNA to separate out our strands. Then we will cool it just enough that primers, because remember, DNA polymerase doesn't add on to just freaking nothing next to it. It has to add on to something next to it. And so we give it primers that will help stick at the ends of the whatever gene that we are interested in. So that whenever we add, we start activating the DNA polymerase, it will add on nucleotides in that area that we're interested in. Okay, so we have separated the strands with the heat, denaturation. We have stuck the primers on there by cooling it a little bit. That's called annealing primers or priming. And then the third part of it is elongation. So just letting the DNA polymerase add the nucleotides on. The nucleotides are just chilling out in a buffer that we have all of this going on in. The buffer also contains stuff that the DNA polymerase needs just to do its thing. Some things like magnesium, that's a cofactor for it. So you also need the template, like whatever you're starting with, my DNA, for example, that sample of the blood at the crime scene, whatever it is. Yeah. And then you need your special polymerase too, but and your primers that you know what it is. The third step, is it not elongation or polymerization? I don't know, the same thing to me. I thought it was elongation. Sorry, I don't have like my books here. I meant to have not the books. I was not ever planning on having the books, but I can open the slide if you want me to, but I'm pretty sure that was just elongation. But maybe we can get the other ones. Sorry, I didn't mean to do that. I guess I can't get out of this. So that was chapter 10. trying to see how it reads this out in this, but I think it was right out here. Extension is the word that this uses. But yeah, that's not as important. I mean, it's more, I don't care that you guys know that the word is extension. I mean, I think if I gave you guys a choice, I'm not going to give you a choice between extension or elongation. Like that's not going to happen. It is not going to be that tricky. So just FYI, but yeah, it's extension on that slide. I was like, where is the slide that just listed them? That was the last one. Okay, so let me go back to here. Okay, get back in here. So, right, so that was PCR. It's not too bad. I like that it only has 3 steps. At least there's that, right? Cloning vectors. The 2 that were like the most, like I say most mentioned, like it talked about it a lot, but there were 2 that we mentioned really. That was the viruses in general, which we had mentioned a little bit how that can work with bacteria. We can also do that with people and just hollow out whatever the genes are that they're carrying so that they're just doing what we want them to do. Another vector that we commonly use in genetic engineering are plasmids. We can essentially use plasmids, cut into them with a known sequence, the restriction endonuclease enzyme, and insert whatever gene we want into it and put that back into, transform it back into bacteria so it'll produce whatever is associated in that, okay? So that's the idea there. Oops. All right, denaturing the DNA. Yeah, we've already talked about that. You can denature DNA either through heating it, like we were talking about in PCR. Heat will dissociate those hydrogen bonds. So that's what's holding DNA together. The double helix is actually held together by hydrogen bonds across from them. The A to the T, that's by hydrogen bonds. So that's what's going on there. So heat breaks up those hydrogen bonds. Those are not exchangeable. Those are not exchanging electrons, right? That would be covalent bonds. They haven't like donated their electrons. That would be an ionic bond. They're just associated by, you know, attraction and polarity essentially. So that is a hydrogen bond. We can dissociate that with heat pretty easily. Same with like proteins, we can do that to them for the same reason. When we're talking about denaturing DNA, we can also denature DNA with acids or bases, because if we have a lot more hydrogen protons or a lot less hydrogen protons than what that thing normally exists in, then it'll interfere with how that they are sticking together with those hydrogen bonds. You know, that makes sense as well. Same with the proteins. So adding something like sodium hydroxide, which if it has a hydroxide, which is an OH, it's going to suck up hydrogens out of this, out of there and lead to low hydrogens, which is high pH, right? So that's basic. So that's what's going to be going on there. So sodium hydroxide is very alkaline, very basic. That's one of the things that I had mentioned in the probe slide. What was it called? Hybridization, nucleic acid hybridization slide. So that one, it talked about adding hydroxide, and that's why, because it interferes with hydrogen bonds and that denatures DNA as well. So it's not just heat that can do that. In PCR, we use heat. We have special machines that just cycle the temperature to do it over and over and over and over again. Restriction endonucleases. Restriction endonucleases was mentioned a little bit just a second ago, but they are enzymes that are normally produced by bacteria in order to protect themselves from foreign DNA, whether that is plasmids that could have harmful, you know, genes in them, or just harmful outside DNA that they never wanted, or viruses, viral DNA. So that's what restriction endonucleases were for. And now we have realized that they are useful for genetic engineering and hijacked them for that. There are a lot of them. They all are something along the lines of EcoR1 or Hind3 or whatever. So if you guys see a letter orientation like that, you would recognize that as being a restriction endonuclease. That's what it means by being able to like recognize it. Okay, you don't need to know them all. Don't memorize them all. I'm not going to have like EcoR1 and then another one, EcoA2, and be like, which one's a restriction enzyme? No, it wouldn't be that. It's going to be like, what type of a, you know, enzyme is EcoR1, and one of your choices would be restriction enzyme or that. So it'll be a little bit more obvious than that. But yeah, restriction endonucleases are used to cut into specific palindromic sequences that read the same on one 5' to 3' as the opposing other 5' to 3'. That way, the interparallel strand, right? And they'll cut at sticky ends so they can stick back either back right together, or they will stick back onto whatever you're trying to put in there that you can cut with the same thing, so it'll match up in the same place. And then you can put all of that you just stuck back together. Of course, you have to do what with it first? You have to seal it up with DNA ligase, of course. Let's not forget it. And you'll seal it up first, and then you can put it into whatever competent bacteria that you would like to put it into through transformation. Okay. I think that's all I want to say about restriction endonuclease. If you guys have any more questions about that, let me know. Oh, that is not what I meant to do. I forgot I wasn't on the actual. I don't know what's happening now. Okay, chapter 24 and 25. These were easy, right? So we talked about the trophic levels, the energy or nutrient movement, that highest energy at the bottom level with the primary producers, and less energy available at the top because we're losing energy the further up we go, like from heat due to metabolism or whatever. So the top guys have to eat more stuff. Yes, EcoR1 is an example of a restriction enzyme. Anything that sounds like that, like EcoR1 or Hind3, or like a few letters with like, some Roman numerals after it or whatever, those are going to be, for the purpose of this exam, readily recognizable as restriction enzymes. Yeah, no problem. But yeah, so high energy at those primary producers, which are photoautotrophs, for example. The heterotrophs of whatever kind is everything past that. The consumers, including the saprobes, are heterotrophs. Saprobes break down the dead stuff, right? They're the decomposers. So all those consumers from primary up to like quaternary was in the picture. The higher we go, the less energy is available. So the highest ones have to eat a lot of the organisms beneath them because the organisms beneath them lost a lot of energy through heat and stuff on the way up that little pyramid. And then basically, yeah, so as a result of that, they'll build up the most toxins. The top ones will, because they're eating more. That's the idea with that. Energy, big to up to tiny at the top, and then toxins are the other opposite way. So biogeochemical cycles, we talked about all the different ones. Think of chunks, and then think of how do we recycle that, stuff. I'm not going to go over like the carbon cycle. You guys should know that one, right? That's just about like the CO2 in the air. And then we have the photosynthesis guys fixing it into like carbon or into whatever organic molecules. And then we eat them, or the saprobes digest them. And as we are using this for energy, we give off carbon dioxide. And then we have combustion and natural carbon dioxide as well. With the water, you guys know the rain and all of that stuff. So you guys are aware of these. Know the basics of them. We talked about cleaning out water, like water safety, how we would test the water, testing for colony forming units for fecal bacteria. If there's general fecal bacteria from us or other mammals or birds, then we're probably not filtering out the really pathogenic stuff either. Potable water, we talked about that. Potable water, and that means just like you can drink it. It's safe to drink. So literally is pathogen free. That's really the only requirement of it. So it's pathogen free. No pathogens in it. We talked about how to clean the water, filtering it out and adding chlorine and everything to kill it. We talked about water treatment for wastewater treatment to pump it back into the river after we pooped in it and whatever, how we treat that, and how we can use microbes to degrade the sludge and everything like that, right? To save more water. And filtering it and then pumping it into the water and all of that. I think that's all we want to say about it. Then we had talked about food and how microbes can help us make food through fermentation, particularly. We talked about beer, we talked about wine, we talked about cheese, we talked about yogurt, we talked about pickles, we talked about sauerkraut and whatever. And we also talked about food safety as far as foodborne illnesses go. I don't need you guys to remember like the organisms that are involved in that. We'll get in that in Unit 4. So don't need to know the organisms. Just be aware of how to keep your food safe, like, keeping it in the refrigerator or frozen, or cooking it completely to kill things off. You know, anything in between, and you might be just helping it grow. I think that's it. So if you guys have any other questions or any specific questions, just let me know. I had originally said I was going to hang out until 3, but you know, I talk a lot. So I'll hang out here for another 10 minutes. So I guess 3:40, so it's 3:53. If anybody ends up having any questions, I will be here. No problem. Happy to help. Yep, no problem. Of course, you guys can always email me too, so no worries.